and stay for the entire duration of the webinar, because this will be counted as your attendance. After the webinar, you'll receive instructions via email on how to claim your PACE certificate. Without further ado, it is my pleasure to introduce you to our speakers today. First, we are joined by Dr. Daan Moiseau, who is a medical immunologist and assistant professor at the Maastricht University Medical Center in the Netherlands. He is an active member of the College of Medical Immunologists, the European Autoantibody Standardization Initiative, and the International Consensus on ANA Patterns Working Party. He has published more than 350 scientific papers in peer-reviewed journals. We're also joined today by Dr. Van Meulen. Dr. Van Meulen has extensive experience in clinical and laboratory medicine and was a professor of rheumatology and internal medicine at the Pontifical Catholic University of Porto Alegre, Brazil. He was also technical director of the Meta-Analysis Center for Medical Diagnosis and Rheumatic Disease Clinic, as well as president of the Brazilian Society of Autoimmunity. He is currently a senior researcher and medical consultant for leading biotechnology companies in the US and is a founding member of the International Consensus on ANA Patterns. Dr. Daan Moiseau and Dr. Van Meulen, we are honored to have you both here today to share your knowledge on ANA testing. And with that, I will hand it over to begin the presentations.
Thank you very much for the kind introduction, and also thanks to Euroimmune to give us the opportunity to share novel aspects of ICAP. Both Carlos and I will present distinct topics in this webinar, and we will alternate several times, as you will see. Obviously, we do have some disclosures, as you can see here from my side, and also the disclosures from Carlos. But having shown that, I like to go to the first part of this webinar, which is about the introduction to ICAP and its goals. And I really want to focus especially on the conception of the idea of ICAP and also on the way it has been implemented in clinical practice. Realizing that ICAP is about ANA patterns, it's a very important step in terms of harmonization of autoimmune serology. And as such, you should realize that standardization of autoimmune serology is quite impossible because every patient has his own repertoire in autoantibodies. But harmonization is possible at several levels. First of all, the clinician has to decide for which clinical manifestation a certain test has to be requested. And of course, that kind of information should also be communicated to the laboratory specialist. Next, the laboratory specialist has to decide which kind of test and which kind of testing algorithm has to be performed. And of course, he again has to communicate with the clinician to tell how things have been worked on. Importantly, then, the test result is to be reported by the laboratory specialist to the clinician. And eventually, the clinician has to interpret these test results in the context of the clinical manifestations of the patients. And as you can see, harmonization of test reporting, as ICAP is about, is the third step of this communication pathway. Now, it was in 2014 that there was a collaboration between the European Autoimmunity Standardization Initiative and the IOIS, and there we had some recommendations for ANA and ENA testing. And one of the recommendations was that ANA immunofluorescence patterns should be reported according to standardized terminology. I emphasize standardized because in the previous slide I mentioned that standardization is not really an option. It should have been harmonized terminology. And basically all people that were discussing these recommendations had a very high score from 1 to 10 for this recommendation, 9.7 with a standard deviation of 0.4. Next, the ICAP workshop, the first ICAP workshop, was organized in Sao Paulo in Brazil. And that was a workshop as a kind of satellite to the 12th International Workshop on Autoimmunity and Autoantibodies. And this was organized by Luis Andrade together with Edward Chen. They are really the founding fathers of ICAP. And following this first meeting, There were several additional workshops all over the world, but especially in Dresden, as you can see. Every second year there is an autoantibody congress in Dresden, and every second year we did have the opportunity to have a satellite workshop about ICAP. And again in 2025, September, there will be the 8th ICAP workshop in Dresden. Now the results of these workshops were, first of all, that after the initiation of ICAP, we made a kind of consensus report. And the consensus report was about the definition of the different patterns together with the harmonization of the nomenclature. And the definition of the patterns was really based on the cell biology. So in the interface cells, you can distinguish the nuclei and the surrounding cytoplasm. And within the nuclei, you can distinguish the nucleoli, and surrounding the nucleus, you have the nuclear membrane. And next to that, in the metaphase cells, you have the metaphase plate surrounded by the plasma. And based on what is recognized in what way, you can define the different patterns that we did in ICAP. And this eventually resulted in the first ICAP classification tree as published in 2015. At that time, we distinguished nuclear, cytoplasmic, and mitotic patterns. We had 14 nuclear patterns, 9 cytoplasmic patterns, and 5 mitotic patterns. We also level reporting, but I will come back to that later in another part of this presentation. Now, going further with ICAP, we did, first of all, some consensus reports, but in 2018, we also defined a negative HEP2 IFA, and we also defined ACXX, a pattern that was not yet included, but it was also not a definite pattern. Carlos will elaborate on that later on in this webinar as well. And we defined an additional nuclear pattern, AC29, which is also known as a topo1-like nuclear pattern. Next, and that was quite an effort, we tried to define the clinical relevance of each separate pattern. And that took us a couple of workshops altogether. I know that we already prepared a lot for the Kyoto workshop in 2016, and we finalized it 3 years later by publication in the Annals of Rheumatic Disease. And that was important because in the first publication in 2015, first of all, we used still the words of synonyms, which is not a kind way of harmonization, because then you want everybody to talk the same language. So you shouldn't use them as synonyms, but previous terms used for that particular pattern. But we also made the patterns to be associated directly with the disease. And we realized in the meantime that the pattern basically is associated with the recognition of certain antigens. And then if you have this combination with it, is associated with a certain disease. So we tried to make structured information for all the different patterns. We decided to have information provided in bullet points. They should be concise, not a complete textbook. They should be evidence-based, so really supported by literature, and they should be useful in clinical practice. So the focus is on the clinician. And as mentioned, there is a pattern antigen association, in particular in the clinical context, which means that if you have a homogeneous pattern and there is a clinical suspicion of SLE, we advise to test for double-stranded DNA. If, on the other hand, if you have a homogeneous pattern in a clinical context of autoimmune hepatitis, it's less likely that you find any antigen, and then we say there is no need for follow-up testing. So that was the idea of the clinical relevance of all the distinct patterns. So the main information is provided at the first level, but there may be also additional information which is presented in supplemental files in the paper and also on the website. And then the information is based only on case series, so this evidence is not that strong, or because the assays needed for determining the specificity of the autoantibodies are not available in clinical practice, but only available in research settings. When we published this paper in the Annals of Rheumatic Disease, we got some comments. First of all, the question was, what about HEP2000? Because at the initiation of ICAP, we decided not to include HEP2000 because these cells were manipulated. They were transfected with SSA60 genes. But based on the question, we decided that we should incorporate HEP 2000, and then we also added some further information on the website for, at that time, AC4, and also for the nucleolar patterns, the pattern that you may see upon transfection of the HEP 2000 cells, if there are anti-SSA60 antibodies present. Another comment was about the clinical relevance of mitotic patterns, because so far we don't know much of the clinical relevance of those mitotic patterns. There are a couple of publications in the literature, but if you look carefully, most of them are somewhat biased in terms of including patient samples with these patterns. Nevertheless, we try to find out if there is any clinical relevance of these mitotic patterns in further on studies that we are performing. The third comment was if the dense fine speckle pattern indeed really exclude systemic autoimmune rheumatic diseases. We still think yes, but there is a but, and that is that this only holds if the pattern is accompanied by monospecific reactivity to DFS70 antigen. So you should have antigen-specific testing together, and you shouldn't find any common autoantibodies against the extractable nuclear antigens. And the final comment that was made, that of course you can increase the relevance of the pattern by also including the titer. It's well known that high titer HEP2 patterns are clinically more relevant in general than the low titer ones. So we fully agree with that. Now next, after publishing these clinical relevance stories, there were some additional workshops. In 2020, we introduced the first ICAP training module. In 21, we had a consensus on how HEP2 patterns should be reported. There were some further reports on the consensus. And importantly, in 2023, we also released the mobile application. And that is very useful, I think, for many people working in the clinical. To get the app on your phone, and the first time that you enter, you really have to register, but next you can use it freely as you want. I think that was a great advantage. And from there, we now have a new latest ICAP classification tree, and as you can see, it has changed enormously as compared. We introduced the negative category. And as you will see, there are also some additional changes. Pattern, we will elaborate on that as well. And the same holds for splitting up AC4 into AC4 and AC31. That will also be further addressed in this webinar. Now, and finishing the first part is realizing what ICAP has really brought to us as a concept. First of all, and that's important in terms of harmonization, we harmonized the pattern nomenclature and also the definitions. We also enabled the coordinated translations in multiple languages, and that, of course, has enabled the application in clinical practice in many, many different countries. Importantly, the ICAP ID was also very well accepted in all kinds of EQC programs, because now we are asked to deliver the results in terms of ICAP patterns. And also the diagnostic industry has helped a lot, because they are using a lot of software-supported microscopes nowadays, and they also try to arrange that according to the ICAP definitions and nomenclature. And recently, there were new recommendations for ANA testing from the EFLM, as published by Caroline Bonroy, and that was also including the ICAP nomenclature. And importantly, as I already stressed, there is a very easy access of the website and the application on your mobile phone for evaluation and reference in clinical practice. And what I show you here on the right is the number of visits of the website over the years. It's cumulative, but you can see clearly that it is increasing over the years strongly. And especially after 2019, when we published the clinical relevance, you see a strong increase in the visits for the website. So I think that ICAP has, within 10 years, very well accepted in clinical practice. Having said that, I'd like to give the floor to my colleague, Carlos, and I shut down my microscope.
Dear Ilana, dear Jan, I would like, first of all, to thanks for the nice introduction, and especially now to Jan about our trajectory with ICAP since 2014, following the steps from the Brazilian national consensus that we started in the year 2000. So my first appearance here is about the novelties of the 7th ICAP, latest ICAP workshop, the educational initiatives, and something about the ACXX pattern. The first project that we addressed in this 7th ICAP workshop was about the pattern study group. So these are separate teams that are dedicated to specific cell compartments and the decoration of these patterns by these compartments by different patterns. And they are coordinated by ICAP members, and the participants are also members of the board committee of ICAP. So, of course, the major aim is to keep the information up to date, not only in our homepage, but in the app that Ian mentioned before. So this is ongoing, and is a big effort because to follow the literature, as you can imagine, is not that simple. The second project we addressed was the HEP2-CIC. It means clinical and immunologic characterization of patterns. This is a worldwide project, and the leader for this one is Luis Andrade out of São Paulo. There are 3 branches for that. Branch 1 is ongoing now. It's about the frequency of ANA patterns reported in labs worldwide. There is considerable heterogeneity in operational characteristics in each lab, each country. And there's, of course, the need for addressing the dissemination and all about ICAP in additional education modules, as I'm going to talk about later on. The second branch is the establishment of clinical associations for new novel patterns, and branch 3 is the determination of autoantigenic specificity, talking a little bit about molecular biology. And of course, for example, we have chosen as priority associations for Golgi, the Golgi pattern, cytoplasmic, and about SG2NA, nuclear antigen that appears in phases S and G2, and nuclear matrix. I have examples here. We're not going to talk about the Golgi now specifically, but it's AC22. So there is mention in the classification tree for the Golgi product. We want to know about the clinical relevance for this pattern. There's some literature about the association with primary Sjögren's syndrome, but that's certainly not enough. This is the pattern for anti-SG2NA that was described in 1994, if I am correct, in Dr. Engten's lab here in San Diego, and the nuclear matrix associated with heterochromatin. This is an important pattern. It's not that rare, at least in my experience, and they are both assigned as ACXX nowadays. They will probably receive an AC number in the near future. As I mentioned, we have educational modules. Module 1 was the introduction to ICAP, and this was performed and finished by more than 4,000 to 5,000 people. Around 1,000 were certified. There is a test at the end. If you pass with some stars, 80% of stars, correct answers, then you receive your certificate. Everything that comes out of ICAP is for free, I should mention. Module 2, we are working on the unclassified patterns, that is to say ACXX patterns, the rare and unusual ones, not fully well characterized. And we'll have the functional anatomy of the heptocell as module 3, and module 4, the compound patterns that we are going to talk today later on. Something about this is ACXS classification. It's an umbrella designation to accommodate a variety of unusual and unclassified patterns that the technician doesn't know, they've never seen. We at ICAP maybe haven't seen or have not well characterized yet with an AC number. And the educational model then will provide some examples and the supporting literature for that. The new box in ICAP classification tree, as Jan showed to us, has an hyperlink to images, and it received a rectangle there at the extreme right. And so it's something that is at a competent level, as you can see here. Some caveats in the ACXX. We already had published in 2018 something about this pattern, and there were some doubts. So I would like to share with you that first, these ACXX patterns are not static. They are to novel AC designations with the number. As much as we can develop new knowledge and assign a number, because we know maybe clinical relevance, we know the autoantigen involved, we characterize the autoantibodies and so on. Recognition of ACXX pattern is important then to prevent their misclassification as a traditional ICAP AC pattern. It always depends, of course, in the situation of the technician, the knowledge of who is performing the text. Substrate quality is very important. We know that different commercial brands of heptoslides behave differently regarding pattern recognition. I mentioned already, training and experience are critical from the side of the laboratory technicians. And number 5, report an XX pattern together with remarks explaining what you are really seeing on the microscope would be my experience that I would like to suggest you, because otherwise the clinician will not understand what is an XX, for example, unless he goes or she goes to the anapatterns.org homepage or to the app. Finally, we decided to create on the Hall of Fame, let's say, for ICAP, and these are the honorary members. We have Dr. Alan Vick from Denmark, and he was the first then to receive this mention for his efforts to the serodiagnostics of systemic autoimmune diseases, especially to immunoassays for autoantibody determination, and promotion of initiative focused on harmonization and standardization of lab procedures, interpretation, standardization of diagnostic immunoassays. You can read here. I read because it's a very important mention about this. The next to receive this honor will be my mentor here in the USA, Dr. Eng Tan, that recently passed away, in recognition of his exceptional contributions to the ANA field spanning 6 decades. There will be a specific session in Dresden now in September for the 8th I2 honor. And all his pupils will be there for sure, and this will be a very important activity. So with that, I come back to Jan to discuss AC30. Jan, thank you.
Thank you. And perhaps adding to your words for Eng Tan, he will be also commemorated in the whole Dresden meeting, so not just the ICAP workshop. But Carsten Conrad also decided to honor him for his contribution to autoimmunity in the whole meeting, which is great, I think. Well, I already mentioned that we introduced two new patterns, basically by splitting up two existing patterns. So one is the splitting up of AC2, where you have an additional pattern AC30, and the other one is AC4, which is split up into AC4 and AC31. Now, with respect to AC30, you should realize that initially we had a so-called dense fine speckled. Now we still have this dense fine speckled pattern, but now we also have this fine speckled with mitotic plate. Both are expert level patterns, and the pattern above is the speckled with mitotic plate with chain. As mentioned before, basically the patterns are defined on how different compartments of the interface and mitotic cells are being stained. So in case of AC2, you see that the nucleoplasm and the metaphase chromosomal plate is positive, and they are both positive in a heterogeneous speckled way. And to be more precise, the heterogeneous speckled pattern in the nucleoplasm is heterogeneous in terms of size, brightness, and density. And because of that, the periphery of the nucleus appears as an irregular and discontinuous rim. And basically, this is more or less the same for the metaphase plate in the metaphase cells. And altogether, the interface nucleus is less bright than the metaphase plate in the dividing cells. Now, here you can see two examples of the typical nuclear dense fine speckled patterns. And I hope you can recognize that these speckles are really heterogeneous in size, in brightness, and so on and so on. And on the right side, you also can see that the metaphase plate is stronger stained than the nucleus of the interface cells. So that are typical hallmarks of the AC2 pattern as we have described it. Now, if you go to the clinical relevance of this AC2 pattern, it's basically, as we discussed before, it is typically found in patients that are apparently healthy or at least do not have systemic autoimmune rheumatic diseases. But this association only holds if you can show that the antibody is reactive to DFS70 and no other common ENA antigens. The third part is that, and that was from the original clinical relevance of AC2, that both in the apparently healthy individuals as well as the patients that do not have this systemic autoimmune rheumatic diseases, the AC2 pattern may be caused by antibodies other than DFS70. And this actually caused the discussion about whether there is some kind of pseudo DFS pattern, which is not the real DFS pattern altogether. And from there, the discussion went forward, and it was decided that indeed you can split up AC2 into the real AC2 that is associated with DFS70 and what we now call AC30, which is not associated with DFS70. And if you look at the pattern, it's different. It's still staining typically the nucleoplasm and the metaphase chromosomal plate, but it's not a heterogeneous speckled pattern, but it's a homogeneous speckled pattern. And that's all over the interface nucleus and also in the metaphase plates. And again, here you do not see such a strong difference in intensity between the nucleus of an interface cell as compared to the metaphase plate of a dividing cell. And on the next slide, you can see the difference. On the left, you see the typical AC2 pattern with a heterogeneous speckled pattern. And on the right side, you see an example of AC30, which has a much more homogeneous speckled pattern. And obviously, it may be quite a challenge to differentiate this AC30 pattern, if it shows like this, from a classical homogeneous AC1 pattern. So there is, of course, always a kind of gray zone in between patterns that we recognize, in between AC2 and AC30, but I think also between AC30 and AC1. And again, that needs training and expertise. Importantly, and that's work from Luis Andrade, he checked this for many samples. And if you typically have the AC2 pattern, as shown here, those are the ones that are reactive almost all for DFS70, while the ones that have the AC30 pattern hardly react with the DFS70 antigen. And the other way around, if you check for other antigens, so not DFS70, AC2 hardly recognizes, but some are positive, of course, but AC30 much more often recognizes the Rho-La antigens or the double-stranded DNA in the samples. So it's less associated with not for excluding systemic autoimmune rheumatic diseases. That is still the case for AC2. Now, if you now look how AC2 evolved in the classification trees that we had, this is the classification tree of 2015. And at that time, the dense fine speckled pattern was still part of the speckled nuclear patterns. So together with the fine speckled and the large coarse speckled patterns. But realize that the difference, of course, is that the metaphase plate, that's given by this sign, is positive in AC2, but is not positive in AC4 and AC5. So the next step was that we said, okay, the dense fine speckled doesn't really fit into these speckled patterns, but should be a separate pattern next to the homogeneous pattern. And finally, now we have replaced this this dense fine speckled pattern by the speckled with mitotic plate combination of AC2 and AC30, and below that you have the expert pattern for dense fine speckled and fine speckled with mitotic plate staining. And having said that, I give the word back to Carlos to tell more or less the same story for AC31, I guess.
Yes, thank you, Jan. There is a story behind AC31 that is the myriad discrete speckled pattern that I'll briefly tell you. So our story starts in 2013, and this was the first description of what was called an anti-SSA Rho pattern. It's a novel pattern at that time, again, from the group in São Paulo, with the leadership of Luis Andrade, one of the coordinators of ICAP until today. And they started to see in 86 samples of from patients this pattern with an association in 99%. It was from the 60 kilodaltons only protein. So this was presented in different commercial slides, and it was published in the Journal of Immunological Methods. In 2021, there was an international multicentric effort to take a look on the same situation. So people from Germany, Argentina, USA, what else? Brazil, of course, they started to see the same. And it was published in Frontiers Immunology in that year. So they again were citing the myriad discrete speckled nuclear pattern and the correlation with the anti-SSA Rho60. Here is from that paper, what they called then as AC4A, the new myriad discrete nuclear speckles, and to your right, the AC4B, that would be the traditional one, the plain fine speckled pattern. So nowadays, just for you to be well situated, you can see between parentheses on top what we are calling now AC31 and AC4. Refining the pattern, there were some experiments conducted by Dr. Chen at the University of Florida with this monoclonal named A7, as you can see, typical pattern in the HEP2 cells. And this figure here is representing that monoclonal. So to differentiate AC4 from AC31, we have in this latest publication from ICAP in Autoimmunity Reviews 2024, what we are now naming AC31 to your left. It's the myriad discrete tiny nuclear dots pattern, and the AC4, what it's called in this publication, is smooth inconspicuous nuclear texture. The autoantigen associations, before you ask, are here in this slide. As you can see, AC31 is specifically associated with the 60 kilodaltons protein, as mentioned before. And from AC4, you can find this in our homepage, is associated with many other autoantigens. So considering the clinical and immunological relevance of this new pattern, the 7th ICAP workshop decided to incorporate it into the ICAP classification tree, clarifying that it is an independent pattern and giving it the AC31 description. So this is the historical summary that you have here, 2013, the pre-ICAP. ICAP was founded in 2014, just for you to remember. It was called SSA whole pattern. Then in 2021, the international collaboration, AC4A, and now at the 7th ICAP 2023, AC30. And here are the synonyms that if you find in the literature for any reason currently in your studies. So there are some clinical associations just for you to subcutaneous, acute, subacute cutaneous, primary Sjegren syndrome, and some overlaps that you can find this anti-SSA whole 60 kilodaltons. This is the site where AC31 is now in the classification tree. It's spread as an expert level, as you can see. It's under speckled AC4 531. Back to Jan. Thank you.
Thank you. And the next part will be about the competent versus expert level distinction and the clinical relevance, how to deal with that. Obviously, we did have is meant by competent, what is meant by expert level. And that was also clearly defined in the recent paper from the last workshop. So basically what we state is that the competent level is considered relatively easy to identify. It should be recognized by all people that perform HEP2 immunofluorescence stainings, and it doesn't necessarily implicate clinical relevance. On the other hand, the expert level is considered to be of increasing difficulty in accurate identification, and any person who should strive to master the recognition of these expert level patterns. Individual samples, however, may be reported at the competent level. So even if you decide to report on expert level or work in your laboratory with expert levels, there are some samples where it's not that easy to distinguish. And then it's no problem to give for that sample just the overarching competent level pattern. Expert level reporting may, of course, increase clinical relevance. Now, coming back to the point that individual samples may indeed be reported at competent levels, that has some consequences if you go to the classification tree. So this is the most current classification tree. And now if you, for instance, consider yourself for a certain sample not to be able to distinguish the new AC31 pattern from the AC5 pattern, you still may decide to report it as speckled, nuclear speckled pattern. And you may give the combination of the AC numbers together with the pattern. Similarly, if you have a mitotic pattern and you think it's difficult to distinguish these, you may use the overarching mitotic name to these patterns. Nothing wrong with that. However, what we didn't think carefully about, and I realized when I was preparing this presentation, is that recently we also decided to separate the discrete dots, the cytoplasmic discrete dots, from the cytoplasmic speckled patterns. So first of all three, the dense fine speckled and the fine speckled cytoplasmic patterns, were grouped together with discrete dots. But now we decided that they should be separated, but there is no overarching competent level designed for this pattern. So perhaps we should in the end decide that the discrete dots are not that difficult to differentiate and could be a competent pattern, but that's something we have to think about. The same holds for the topo 1 pattern, AC29. Again, there it's a nuclear pattern which is might be quite difficult to recognize because you have to recognize different parts of the cells. But there is no overarching competent pattern that is covering this expert level pattern. So that is a challenge, of course, that we have to take into account for future classification trees. I think people that nowadays are not able to differentiate the topo 1 pattern, they just report it as the homogeneous nuclear pattern. Having said that, we have to realize that several patterns have different clinical associations. There are some patterns that have a rather high clinical association, clinical relevance. In particular, the centromere pattern is very clearly associated with the centromere protein antibodies, and as such also with systemic sclerosis. And if you look in the classification criteria for systemic sclerosis, it's sufficient to have this pattern. I think that's the only exception. For nuclear dense fine speckled, for reticular antimitochondrial antibodies and the topo 1, they are also of high clinical relevance, but then it requires that you identify the correct antibody antigen specificity. So for the nuclear dense fine speckled, it's a DFS70. For the reticular AMA, it's the pyruvate dehydrogenase E2 component of the mitochondria. And for topo 1, it's of course topoisomerase 1. Next to that, on the other side, we also have several patterns where we don't know, or at least perhaps there is no clinical relevance at all. That holds for instance, for the few nuclear dots, also for the smooth nuclear envelope, for the Golgi. Carlos already mentioned that we are searching in studies clinical relevance, and the same holds for the different mitotic patterns. And obviously, there are a lot of patterns in between that, depending on the clinical manifestations and the antigens that you find, they may have more or less clinical relevance altogether. The important thing is that the clinical relevance so far has been defined based on all the expert level patterns, so not for the competent level patterns. And what I mean by that is that, for instance, for the nuclear patterns, we have 3 expert level patterns, and they each have its own clinical relevance. If we look at that more carefully, you can see that AC8, the homogeneous nuclear pattern, is associated with systemic sclerosis if you have these types of antibodies, or if you think about these diseases, you should at least test for these antibodies. AC9, the clumpy nucleolar, is also linked to systemic sclerosis, and if there's a clinical association, you should check for the fibrillin antibodies. And the third one is AC10, the punctate nucleolar, and again, that you also find in the similar types of diseases, and there are some additional antibodies. So even if you use the overarching competent level pattern nucleolar, you still have to search for autoantibodies associated with systemic sclerosis. And basically, you do that normally in a panel assay where you have multiple antigens for this typical disease. However, this is different if you consider the nuclear dot pattern, because there we have multiple nuclear dots, which have clinical associations in relation to PBC, juvenile dermatomyositis, and some inflammatory conditions. And then depending on what kind of clinical manifestation, you should search for other antibodies. But if you have only few nuclear dots, there is very little clinical association, no need for further testing. So if you put this all together, basically you have to follow the clinical relevance and the follow-up testing advices as defined for AC6 only. And essentially the same holds for the dense fine speckled pattern. So the new AC2 pattern, there you should search for DFS70 antibodies. But now we split them, and we also have the AC30 pattern, and in that pattern it's not of added value to search for the DFS70, but you are encouraged to search for other antibodies, because you may find them depending on what kind of clinical manifestations are reported. And that, of course, brings us to several challenges for ICAP. First of all, we need to increase the awareness among clinicians. Sorry, that's not the idea. We need to increase the awareness among clinicians of the clinical relevance of the different patterns. It was questioned by Lieve van Hovels a couple of years ago by questionnaires sent to laboratory specialists and clinicians. And at that, there too much of the clinical relevance of many patterns. Centromere pattern, for sure, but beyond that, it was very limited. Next to that, we also have to find a way to keep up the clinical relevance, to keep it up to date, because new publications will be published. There will be new information, for instance, about Golgi or mitotic patterns, and we have to incorporate that into the website. And eventually, I think we also have to find a balance between extension to more patterns, as we have done now for AC30 and AC31, but also the balance with the further implementation in the clinical practice. Because people working in clinical practice have to keep up with the speed of new patterns and to get used to that as well. And hopefully we can achieve that in the forthcoming workshops. And from there, we can go on back to Carlos again for the compound patterns.
Thank you, Jan. Let's take a brief look on compound patterns. This could be quite complex. I'll try to simplify it for you. So if you have more than one pattern on your immunofluorescence assay, by your feeling, your knowledge, you are in the presence of compound patterns in opposition to a simple pattern, of course. And this is the staining of a single cellular domain. I have one example here. Jan just mentioned the nucleolar patterns. This is typically found in the scleroderma, but not only. And it's one cellular compartment that is really with a strong fluorescence. In opposition again to that, we have the compound patterns that we arbitrarily named at ICAP as composite, mixed, and multiple. But definitions for compound and composite. Compound is something made of multiple parts, made up of two or more separate elements, often distinct or separable. And the easiest way that I found is to mention to you water, right? So it's H2O. H2O is a chemical compound. So this is compound. Now, composite is different in the English language. It's something made by blending multiple elements into a unified whole, often with a sense of integration. And then I remember that there are some swimming pools regarding water that are made of fiberglass. And so this is what we can then have a feeling of what a composite is. Using water, but it's different, right? So the compound patterns for us at ICAP are first the composite ones, staining of different cell compartments by the same autoantibody, one autoantibody only. And this is the example that I have here for you. Anti-TOPO1 in the past was anti-SCL70, as you know. You have nuclear, nucleolar, and cytoplasmic decoration is only one autoantibody, different cell compartments, and you have other examples in the square at your left, like AC14, 26 also are in the same category. Mixed is the co-occurrence of two or more simple patterns staying in the same cell compartment. Normally, this comes about on the nucleus. And you can see here an example of the AC1, an homogeneous pattern. Of course, you are going to have to titer out. This is very strong decoration in this experiment, and then maybe we start to see some AC4, AC5, and this would be anti-SSA60, Ho60, and anti-RNP, ribonucleoprotein, U1, that will appear also in the nucleus, and then you have the mixed pattern in this combination. So it's 2 or more autoantibodies involved. And the third classification is multiple. Multiple is single plus single. 2 or more simple patterns in different cell domains, as you can see here. Anti-P95, sometimes called PA100 or S100, and the mitochondrial cytoplasmic pattern. This is then 2 cell compartments. And what you have here are 2 different autoantibodies. One identify an autoantigen in the nucleus, the other one identify this network of mitochondria in the cytoplasm. So normally it's an easy independent recognition. Here you have, I'm not talking about this right now because it's just a summary information for you. You can maybe make a picture here, or this will stay in the homepage for you. And after our seminar, and you can have the feeling what is simple, what is compound, the cell compartments involved, how many autoantibodies, and you have examples in this table too. So what's the importance of compound patterns? First, classification is arbitrary, as I mentioned to you. It's important for the lab technician and for research, more than for clinicians. As a clinician, as a rheumatologist, I think that the description of these compound patterns do not add much to my care for the patients. For example, I don't say to the patient, drink H2O, right? I say, drink water, 1.5 liters per day, something like that. So I go directly to the real information that patients can understand. I also don't tell them, look, you have like a spondyloarthritis, you must swim in the swimming pool. I don't say, use your fiberglass container once a day. I go to the real information to the patient. So for me, it would be most important that we know the clinical relevance as our publication from ICAP with the leadership of Jan in the past. And these are the important steps, and not differing for the patient exactly the way these compound patterns are elicited and described by ICAP. So this is from the clinical side. That's my take. Back to Jan with the key takeaways from our presentation.
Thank you, Carlos. Yes, what did we learn, hopefully, today? First of all, I think that we should have learned that ICAP provides harmonization of pattern nomenclature and definitions. And it is important to realize that this is widely accepted in clinical practice, and therefore it's also important that we all use the same nomenclature and definitions, because communication, of course, is key to patient care. Second, ICAP is active in spreading the information on HEP2 patterns, and we use the website, but also mobile apps. We have teaching modules, presentations, and publications, and basically everything is freely available. The only thing is that you have to do is to register on the website, and then you have access to all these applications as you want it. And you can also spread the news, of course, to your colleagues. Third, the HEP2 classification is not a static system. I hope that I showed you that the classification tree has changed over the last 10 years several times. And that is because we learn, we have discussions at different workshops. And again, we will have a workshop in September again. So, and we are depending on the whole society that is working with HEP2 to give us information and to tell us what is needed in order to further improve our classification. And finally, the novelties that include specification, the novelties that we showed include, and also the definitions for comp. And before closing, I give the last word to Carlos again, and he has some final remarks.
So I would like to thank for your attention, for the invitation from Euroimmune, for Jan to be with me here. Our friendship runs for many years, and it was my pleasure, my honor to be with him here again in this scientific forum. I would like to say that we need your help. You that are hearing us today, watching us here, the sustained feedback that you can provide is essential to improve ICAP in our mission. So we really need ongoing systematic updates. And if we need at ICAP, you will have the possibility of receive all this information in our web page and our app later on. So it's very important to keep up to date. There's lots of information around. And I would like to remind you this famous philosopher Montaigne from the Renaissance. He said, what men do not understand, they classify. So that's what ICAP is doing, right? We don't understand autoimmunity in its totality. We don't know many things where we are still ignorant. Maybe AI comes to the rescue, but it's very important that we stay and we strive to find the truth in each sample that we examine in our feedback. What are the unmet needs, additions, suggestions that you have for us at ICAP? And with that, I thank you very much, Jan, again. And I thank you, Ilana and Euroimmune, and back to Ilana for finishing the seminar. Thank you very much.
Thank you very much to our speakers for those insightful presentations. We're now going to open the session up for a Q&A session. And thanks to those who have already asked questions in the chat, there's still time to enter questions, so please continue to do so in the chat box. I'm going to start with the first question, which was asked by a few people regarding the recommendation for ANA dilutions. Our speakers answer that.
I can start with that. Yes, there is a lot of discussion, of course, which kind of dilution should be used. There are many people, if you look in all kind of surveys, that most laboratories use a 1 in 80 or 1 in 100 dilution. There is also a discussion whether you should use a lower dilution for children. There was a recent publication actually from the EFLM, and there they showed that there is not real evidence that that is necessary for children. So you can use the same dilution for adults and children. But of course, if you use a 1 in 80 dilution, it may be fine to see the pattern better, but the clinical relevance is far less. So I know work from Xavier Bossuit. He showed that those lower dilutions have likelihood ratios close to 1. Which means it doesn't add much to the clinical decision of the patient. The higher the titer, so 1 in 160 or even 320, the more clinical relevant your result is. So altogether, I think a starting dilution of about 1 in 80 is fine, but clinical relevance starts higher up in the titration phase.
May I add something? At 1 to 40 that many labs here in the US still use, I would like to remember people that are watching us, we can reach false positives around one third of the samples. So that's not exactly what the laboratories want to offer to their clients, the physicians. At 1 to 80, we go to 13% more or less. 1 to 160, even lower, And the clinical decision limit for me as a clinician is 1 to 320. Then I'm on solid ground. I know that this is really important as a result, and this is what I have to find in the samples of my patients if I want to offer, if I am in the lab, and offer the clinicians the best possible valuable result for them. So I agree with Jan. Normally we start at 1 to 160 even, and maybe there are some labs starting at 1 to 320 now. But you must decide on your own situation and according to your local population, of course.
Thank you so much. I'm going to move to the next question, which was asked also by several people about the mobile application, the ICAP mobile application. Can you talk more to that and also speak to its availability? We had a question about whether or not it was available in Ireland or other countries.
Yeah, well, first of all, I think it's important to realize that if you want to use the mobile app, you should first register and become an affiliated member on the ANA website. So that's the first thing you have to do, because then you also have login codes. And next, you can go to your app store, whether it's on Android or Apple, and there you should search for the ICAP official app. And you can download that, and then you have to log in the first time, but all consecutive times you don't have to log in anymore. So that makes it much easier. There is this discussion whether it should not be available in some countries. We checked that with the person who devised this mobile app, and he confirmed that it should be possible to download this app basically in all countries, except perhaps for Italy. There may be some restrictions there, but there should be no restriction for Ireland whatsoever. And if persons have problems there, they might contact us and see whether we can solve the problem, I think.
Thank you. Next question, which we were asked in several different ways about how do we interpret a negative ANA IFA pattern where then we get a positive on a solid phase assay test, such as the example given here was an ENA 23 panel with a patient showing symptoms of autoimmunity. How do we interpret such negative ANA IFA results?
Start with that, Jan, if I may. It's not that rare that this situation occurs. It's important to know that the substrate that we use, the HEP2 cells, is coming from a larynx cancer in a patient from the mid-1950s. So this is an immortalized cell line, is an epithelioid cell line, human epithelioid cell line. And so it's not enriched in many of the autoantigens that we need to, let's say, discover what is happening in one's serum. So, for example, it's HEP2 is notably for not having let's say, the real right amount for us in the lab to detect anti-SSA, for example, or anti-SSB, also in the process of doing the fixation and permeabilization protocols. These are very small molecules and they are extractable. So that's from that situation came the term extractable nuclear antigens, right? So, but we have other ones, like the Jo1 can be negative. It's cytoplasmic, of course, but you can have high titer of ribosomal INP by ELISA and ANA negative also. I have 2 or 3 examples from my lupus patients. So one must be aware that this is a possibility and this is a technical problem we cannot solve.
And perhaps I may add to that, because there are also some questions about row 52. Altogether, row 52 does not give any HEP2 pattern. So that's completely irrespective of the brand that is being used. So that's one thing. Carlos already mentioned that Jo1 may be missed, but that holds for most myositis-associated antibodies, that the sensitivity of the HEP2 cells in general is too low. So it's indeed very common that you may have a negative ANA HEP2 result in combination with an antigen specificity. But it depends on the specificity on how you have to deal with it. Because I would be a little bit nervous if you see a discrepancy in terms of the centromere pattern and the centromere proteins. That would be much more unlikely to occur. So then I would really check for additional tests to see whether you have the right test results. So altogether, it depends on the antigen specificity and to a certain extent also on the brand that you use for HEP2 cells. And if I may extend on that, because there was also a question on the type of brands, what ICAP is doing about that. We know that there are differences in how antibodies react with the HEP2 cells from different brands, and we are trying to make a kind of show these differences. The rods and rings pattern is most typical for being well recognized in some brands and totally not recognized in other brands, but you also may see differences in patterns for other autoantibodies. So that's something we are working on, which is not yet finished, I have to say. But that's the story. Thanks.
Thank you. We got quite a few questions now about testing and confirmatory testing and what testing should be done to confirm these antibodies. One was, should specific autoantibodies be tested and ENA screening performed to differentiate between AC1, AC2, and AC30? So what would you recommend there? Differentiate between those patterns.
Yeah, well, that's a good question, I think, because especially those patterns may be difficult to distinguish, especially if they are mixed. So you can imagine that you may have a homogeneous pattern together with a AC30 pattern, and I think it's almost impossible to discern those two types unless you have different titers. So if you do a titration, you may see the difference. So in those cases, it depends a little bit where you are working, because in some countries it's free to add additional tests for finding the antigen specificity without requesting the clinicians first, while in other countries you may only suggest to do these kind of tests to the clinician, and they have to order it before you can do it. But altogether, I think in those situations you to a certain amount, you depend on the clinical information that you have. So, for instance, if you have a patient with autoimmune hepatitis, we hardly can find the antigens because they were never defined. But if it's a patient suspected of lupus, of course, you have to test for a double-stranded DNA antibodies, and so on. So it depends a little bit on the algorithms that you are allowed to use in combination with the clinical information that you have. If you don't have any clinical information, I would at least suggest to advise the clinician to further test for a different set of autoantibodies.
I would like to emphasize, and I would fully agree with Jan, the clinical side is very important in this kind of situation, and there should be a communication channel established, like in one of the first slides that Jan showed us, because otherwise we can run into problems. So good communication, lab, physician, lab, the clinical side, and we should remind people that are here with us, one test like ANA doesn't make a diagnosis in the vast majority of the cases. You need the clinical side to correctly interpret the results of this test.
Thank you. The next question I'll ask is, I believe based on what terminology to use, they mentioned ANA versus anticellular antibody reporting. maybe they're asking about, what terminology to be used, and also maybe we can then speak to, is it important to report patterns that are not only nuclear but cytoplasmic, etc.
And I think I can start with this one because it was a long discussion during the early years of the Brazilian consensus. We started in the year 2000, as I mentioned before, and ICAP started in the year 2014. And we had the discussion because exactly the term anti-nuclear antibodies is the wrong term for this test we know today. So we are reading more compartments than the nucleus. So in Brazil, we devised at that time one possibility to read all the patterns as positive, including the cytoplasmic patterns, maintaining the name of the test, anti-nuclear antibodies. So the way we devised that was to add a subtitle. And the subtitle was anticellular antibodies or anti-cell antibodies like ICAP took thereafter. So even in our report paper, How to Report ANA from 2021, we added this. And it's very clear-cut the way we think one should report the test. We would say anti-nuclear antibodies by immunofluorescence in hepatocells cells, for example, and just after that, in parentheses maybe, anti-cell antibodies test. So, and this was the basis for the classification also for anti-cell 1, 2, 3, 4. So it's very important that people know the best way to determine the name of a test is the best way it describes the test. And this is for historical reasons and economical reasons. We cannot change this test because most of the coding for the lab to receive money for this test is based on the old terminology with these codes. This happens, I guess, in almost all the countries. So that's why we are not pushing forward with the new name, just naming it anti-cell antibodies. Then you could say a nuclear pattern is anti-nuclear positive. You could say a cytoplasmic pattern also is anti-cell positive. So this is important, but we cannot solve the situation due to these grounds.
Yeah, and adding to this story, we have to realize that cytoplasmic patterns may be clinically relevant. That's something we shouldn't forget. So if you only focus on the nuclear patterns, you are missing clinical relevant information. So especially if you, of course, look for the anti-mitochondrial antibodies, they give a typical cytoplasmic pattern. Still, you have to confirm that with antigen-specific assays, but that is a very strong link to a disease which is called PBC. Next to that, you also have the AC19 and AC20 fine speckled cytoplasmic patterns, and they may be associated with the what we call the anti-synthetase antibodies related with myositis, and also the SRP antibodies may be related with myositis. So if myositis is considered and one finds such a kind of cytoplasmic speckled pattern, it's really important to report it to the clinician. However, we should keep in mind that the myositis-specific antibodies, as we discussed before, might be missed on the HEP2 cells. So previous ENMC recommendations said that if there is a clinical suspicion of myositis, perhaps it's better to go directly to the myositis-specific antibody multiplex assays. But nevertheless, those antibodies are definitely clinically relevant in general.
Jan, sorry I didn't send you specifically this slide. I prepared this one exactly when I give these classes on cytoplasmic antibodies. And you can see here there is summary information from AC15 to AC23. Then we don't have all cytoplasmic patterns here represented because they sometimes really do not have a relevant clinical association. That's something that we are exactly now studying for the polar cytoplasmic that represents anti-Golgi AC22. You can read that as rare in Sjögren's syndrome. It means that there is a publication, one or two, about it. But we are not happy with that, so we are going to try to study this in much deeper. But here you have some information of what Jan just told us, and you can have this as like a reminder for the ask you, why are you reading cytoplasmic patterns now?
Thank you, and thank you for this resource. I'm going to ask now questions about patterns specifically. We have quite a few questions about patterns. Someone asks how to differentiate between an AC31 pattern and sort of a mixed pattern of a speckled with nuclear dots, maybe. Speak to maybe a way that someone could differentiate between AC31 and a mixed pattern.
This may be a difficult situation in practical clinical situations. So, yeah, I think this adds to the situation of the competent versus the expert level. So you need time to be able to really differentiate what is myriad discrete from what is plain fine speckled, as we define in this latest publication. There's no other way than just to have the feeling and have a mentor maybe, and see the many examples that we are going to have in the homepage and in the app. We are sorry that we still don't have this info ready to go for AC30 and AC31 in our homepage, but we are selecting the best pictures and trying to find the best way to teach people how to differentiate. Please be patient. It is coming in the incoming weeks. Jan, would you like to add something from your perspective?
No, I think it's a clear point what you made here. Yeah, fully agree. And I think the further information on, because that was also a question indeed about when will the information be available on the website for AC30 and 31. Perhaps we have a further discussion in Dresden with the ICAP team, or a little bit before. We are trying to arrange that, but then this should be finalized. We are circulating the documents basically in these days and see what we can define. We have it in the right context on the website.
Great. And to follow up on that AC31 pattern, we had a question about its clinical association. Would you consider a high association between this new pattern, this AC31 pattern, and Sjögren's syndrome?
Yeah, for sure, because it represents the anti-SSA rho 60 kilodalton. And this is, in reality, present, let's say, 60 to 80% of primary Sjögren's syndrome patients. And it's an autoantibody that is associated with a more severe disease, with vasculitis in Sjögren, with more Zika symptoms, and including lymphoma in the long run if the patient is not treated well. So it's one of the important clinical associations, and we must describe it.
And I think it's important to realize that SSA 60 is not specific for Sjögren's syndrome. You see it quite often also in SLE, especially in cutaneous forms of SLE. So be aware that it's again to be interpreted, and that's all the time the same story. It's to be interpreted in the context of the clinical manifestations of the patient. So I think that is important. Also important is to realize that SSA 60 antibodies react differently in HEP2000 cells. So there again, you may have some difficulty in distinguishing these AC31 patterns as such. It may look completely different because of the transfection. But there is some information on that also on the website.
And that's why ICAP is not dealing with the way that these cells, HEP2000, show the different patterns for the technician. So even the Brazilian consensus in the early days, we did not take into account this. We didn't advise against, for sure, but we couldn't elicit the patterns as such when using the standard HEP2 cells, let's say. So let me just add one historical point, Ilana, may be interesting for people that are watching us. SSA came from the terminology. SS is Sjögren's syndrome, of course, A and D. And this came from Engten's lab in La Jolla at the Scripps Research Institute at the time. And the same serial was exchanged with another pair or some serial from Oklahoma, from Dr. Morris Reitling. At the time was describing the same autoantigens and autoantibodies that he determined to be Rho and La, that should be the first two letters of the family name of the patients. So they exchanged serum, those two scientists, and they realized they were exactly the same. They were describing the same phenomena. So that's why still in the literature, you have SSA, you have Rho. And normally, people that come from San Diego, from Dr. Ten, named SSA-Rho, SSA first. People that come from Oklahoma or the East Coast, name Rho-SSA. So you'll see that in the literature, but it's the same 60 kilodaltons antigen.
May I expand a little bit on this, because there are two additional questions. One is about the Sjögren's syndrome classification criteria, because the original ones that were published in a couple of years ago, in 2012, they didn't differentiate between SSA60 and SSA52. SSA52 actually is so Rho52 is a completely different antigen. And the reason why they didn't differentiate between these two antigens is because the populations where these classification criteria were defined on were not having this information. So in the cohorts that were included, the distinction was not made. And therefore there was no evidence to do that. But later on they published that it really should be SSA60 and not Rho52, which is a very common autoantibody in many different diseases and also in healthy people. The other thing that was questioned is, can you have ANA positive patients with SSA antibodies but ANA negative? Yes, that's possible. That's exactly the reason why the HEP2000 cells were made, because then you can increase the sensitivity for SSA. But even HEP2000 doesn't pick up all the SSA antibodies. So even those samples with a negative result may be related with SSA60 antibodies. The point is that we don't know, as far as I know at least, if SSA antibodies that are not picked up by HEP2000, if they are clinically relevant. We also don't know exactly if those ones that are picked up by HEP2000 but not by regular HEP2 substrates, if they are clinically the most relevant ones. So such studies are lacking, but the point is that, of course, there is quite a discrepancy between HEP2 IFA pattern positivity, let's say, and the finding of SSA60 antibodies. Thank you.
Thank you. Next question. In 2019, ICAP recommended that, with the possible exception of centromere, all patterns should be confirmed then by an immunoassay for a solid association with autoimmune disease. Is this still a recommendation?
Yes. It's quite simple, because the pattern never gives you full certainty about the antigen that is recognized. And the only very strong association is there for the centromere pattern. But for the other ones, you really, even if you find, for instance, an AC29, so the topo 1 like pattern, you have to define it. If you find the AC2 pattern, you have to define it as being reactive against DFS70. So yes, it's always the combination that you need, for sure.
Agreed.
I'm going to ask now a question that we've gotten in a few different ways about expert level classification, how to deal with expert classification, and will ICAP modules in the future be training on how to, differentiate between expert level, competent level patterns? What's sort of to come as far as the educational modules?
Well, go on, Jan, with this one.
Yeah, there.
Please.
For now, we only have one educational module on the website. There is one almost ready, and that is the one that Carlos presented. That's about the ACXX and that kind of stuff. And definitely there is on the program that we want to have additional modules, and one of them definitely is the distinction between competent and expert patterns. But realize in the end, it's not always possible in every sample to differentiate in the expert level patterns. And then it's no problem that you go back to the overarching competent pattern level. So it's you can freely go in between them as well. But yes, we will try to make a module, but realize that making these modules is an enormous amount of work, I have to say. And those people that are doing this do it for free, basically. So they have to do it in their spare time, and that's the main reason why it takes quite long to have these modules available.
Thank you. Next, we have just a few more minutes. I'll ask maybe two more questions. One is there a consensus on which ENA must be tested for and found negative if there's a positive anti-DFS70 pattern to indicate that the patient does not have an autoimmune disease? So what's sort of the follow-up testing, I suppose, after anti-DFS70 pattern positivity?
Well, the story is that at least you should check for DFS70. But the point is that it should also be monospecific. And I think from ICAP perspective, we never defined what monospecific means, except for the fact that we say it should be at least negative for the most common autoantigens. And that is basically the double-stranded DNA, the SSA, SSB, RNP, SM, centromere, SCL70, and JO1. I think that's the main category that should be checked for. But of course, people may have other thoughts about that. But ICAP never made a complete list of which one should be excluded as being negative.
And it will depend on the clinical side.
Sure.
Thank you. Next, I'll ask a pattern question regarding also unclassified patterns. So someone asks if the pseudo PCNA pattern would be an example of ACXX. If they see this pattern, do they report it as ACXX? Asking.
Yes, you can go with that classification because we don't have an AC pattern assigned so far. I mentioned one example, SG2NA, and we are studying this deeper right now and see even clinical associations. We know the autoantigen involved, but this is something that so far we cannot cover everything there is out there. But it's our intent along the next years to give more and more definitions. So less and less you that are watching us will need to classify unidentifiable patterns as ACXX. That's one of our aims, of course. But as ACXX, It's difficult and it takes time. It takes lots of human effort to be able to put a number in our classification tree. But rest assured, I'm sure that the committee is doing a very nice job in trying to better understand and better give you the best possible classification that we can.
Thank you. We've now reached time. If your question was not answered, we did receive a lot of them. We will respond over email. I want to thank everyone for joining, and thank you again so much to our speakers for joining us. Please look out for a follow-up email for a survey and also instructions on how to get PACE credits and PACE credits. Click on the button that you see on your screen. This webinar will also be available to watch on demand using the same link, and it might take 24 hours in order for you to be able to watch it on demand. And also in the future, look out for additional emails to promote our next webinar, which will be happening in September. I wish you a great rest of the day wherever you are in the world, and we'll see you next time. Bye-bye.
Bye.